Well, thank you so much for the opportunity and for the invitation. It's a real honor to come and speak to a group that knows so much about animals and in computing and managing large data sets. The animal scientists have been historically known for their ability to analyze a lot of very complex data, so I am thrilled to be here just to describe some of the work that we are doing in plants, more specifically in corn. So what I'm going to focus on today is just some of the work that we have been doing in high-throughput phenotyping and how we are working towards applying some of these technologies, as Jim mentioned, to the genomes to fields, which is an initiative that started a few years ago, trying to increase the efficiency or increase our ability to do a better job of predicting phenotypes. So I have to brag a little bit about my species of preference. The difficult thing and the beauty of working in an organism such as corn is that corn, it is really an incredibly efficient plant in terms of using the resources that are around to create important products. So if you think about an average farmer plants about 30 kilograms of seed per hectare in the United States, and there are large numbers of millions of hectares planted in the United States every year. 75 days later, those 30 kilograms of seed have produced, on average, 12 tons of biomass, dry biomass, in those fields. And if you wait another 45 days, you have an additional 12 tons of material that are produced in those fields in the form of grain. So it's an incredibly efficient machine. But it is interesting to think about where that efficiency comes from. And so when we think about the phenotype, in this particular case, yield, we know that it is composed of the genotype and the genetic code of a particular individual. Technologies such as the ability to sequence genomes in very cheap ways have allowed us to really understand a lot of this complexity of how genes express themselves and how they interact. And because of the cost of sequencing keep dropping, there is really an unlimited or a very large number of resources that allows us to understand that component. We also have an enormous access to environmental information that allows us to actually classify environments, to understand how to better place particular genotypes in the environment, and evaluate how to make the breeding process more efficient for specific areas of geography. But then there is this component, which is really the most complicated one, which is the fact that plants are in the environment, and they are exposed to that environment, and they respond for the most part in somewhat predictable ways. In here I'm showing an environment where you have, an environment that has high stress. Genotypes will tend to perform poorer than those that have low levels of stress. However, there are situations that are a lot harder to explain, and we call that particular effect of the sort of the unpredictability behavior of certain individuals in the environment, genotype by environment. And so one of the things that we focus a lot of energy and resources is to try to understand how, when we correlate a particular trait between two environments, that correlation is not always perfect. And if we start thinking about that correlation, not just from a statistical standpoint, but actually trying to dissect what is the biology that causes those different responses, we can maybe do a better job of predicting how certain genotypes will behave in certain environments. And these erratic behavior, unfortunately, is more the rule than the exception. That's job guarantee for every plant breeder out there, by the way. So the system is complex. Nevertheless, breeders have been extremely successful in improving maize. I wouldn't be a true maize breeder if I didn't stand up here and show you a graph of the improvement in yield in the United States for the last 100 years. So this is the most probably shown figure by any corn breeder that you've ever met. So the remarkable improvement in yield, average yields in the United States since the late 1800s to today are remarkable. And the other thing that I want to point out is that if you look at the difference between the distribution of points, even 20 or 30 years ago compared to today, the ability to tighten up our ability to predict with very few exceptions, such as rough years like 2012, is really remarkable. So now we are not only making these genotypes more efficient in producing energy, yield, but we also are doing a better job of making sure that they are consistent in their performance. However, this comes as a cost, and every breeder knows that. It takes a lot of resources to maintain this improvement. There is a clear law of diminishing returns. So breeders and corn scientists are really looking very hard as to how to better utilize the next set of resources and tools to continue to maintain this level of improvement and this ability to really increase performance in a consistent way. So this is the way we used to do phenotype, and a lot of our phenotype happens that way. We take a lot of students out to the field and have them measure everything that we can in individual plants. This is really not scalable, it is not sustainable, so we have to move away from it, and that's what I want to focus on for the most part today. So how do we use high-throughput phenotyping and new technologies to increase the efficiency in plant breeding programs? So sort of the low-hanging fruits are tools that allows us to just improve the efficiency of the method itself. For example, one of the differences that I think are somewhat obvious in terms of plants and animals is that plants, thankfully, they stay static. You put them in a place and they stay there, which is really nice. The issue is that they stay there and you have to go to them to measure them all the time. So what I'm showing you here is a typical experiment that we have where we might have tens of thousands of genotypes in little rows. And I am a pretty fast walker, but it takes me about 45 minutes a straight line to go from one end to the field to the other. So if you want to measure anything systematically in a particular field, any method that would allow us to improve that particular component of the process will be very efficient. One of them that we are trying to implement is actually count the number of seedlings that are actually germinating in a particular field. So if we use UAVs or little rovers that can ride through the field, you can identify where particular seedlings are and actually track their growth through the early parts of the season. The other very important task that we have as breeders is to understand the maturity of our materials, to understand how to best place them in particular places. And for that, we have to walk up and down the field many times to measure flowering. Look at when a tassel starts shedding pollen or a particular female inflorescence puts its silks out. So we walk the length of the field back and forth many times. If we can use UAVs to detect, for example, the presence of tassels, as highlighted here, and you probably cannot see in the back, that will save a lot of time and therefore resources for a breeding program. But that is just increasing the process itself. High-throughput phenotyping technologies can also help us understand different dimensions of a particular phenotype. When we go out to the field, one of the traits that are important for breeding corn, farmers are continuing to push the timing of planting for a particular varieties to increase the cycle of that plant and therefore increase productivity. So selecting corn plants for early vigor in cold soils is very important. But we go out to the field and you look at day number 1 and you see nothing, and day number 2 and you see nothing, and you keep looking and nothing happens. However, if you could look at that same set of materials from a different perspective, and if instead of looking at the growth of the early seedling in the soil, specific tools allows us to see the time of initiation of radical growth or seminal root development. In this particular case, for example, we can start identifying differences in the performance of particular genotypes that gives an indication of what are the most likely genotypes that will be useful for selection. And this is work that we have been doing in collaboration with Edgar Spalding at the University of Wisconsin-Madison. So what other things are important to improve the ability to increase the efficiency of breeding programs? And one of them is if you can accelerate the time when a breeder understands the important traits that will control productivity, that certainly will increase efficiency. So one of the things that we are working very hard is to identify traits early in development that will be indicators of final potential. And that is important also because, as we know, a particular variety will have potential for yield, but it will be affected in very different ways depending on when environment creates specific stresses. So what I'm showing you here are the growth development stages for the corn plant and how specific stresses can have a smaller or bigger impact in terms of the overall productivity depending on when that particular stress occurs. For example, if you have hail or significant drought in the period of flowering, that will have a much bigger effect on the ultimate performance than if it happens otherwise. And this is important not only in terms of making decisions of what to select for, but these are tools that can also make a big difference in terms of managing particular fields and making predictions or making better decisions on when to increase the utilization of certain inputs or different management treatments. So we have been spending quite a bit of time trying to identify what are the traits that would allow us to increase our ability to detect performance earlier on. You're going to have a presentation tomorrow by Liang Dong from the Iowa State University, and he's working on sensors that can actually detect nitrogen flux within the plant. So this idea of a Fitbit per plant, we really think about that. We can, if we can, as Jim mentioned, put a Band-Aid in each plant that will measure how nitrogen moves in and out of a plant, that would be a huge advancement. In the meantime, what we are doing is utilizing image to look at our fields from the top and look at how particular either visual characterization or reflectance of certain light can provide input in terms of plant development. So what you see here is work being done by Nathan Miller at the University of Wisconsin. And what you see, each one of these lines of plants is a row of corn, a different variety, and it looks red, not because we love Wisconsin and we think red is the color to be. It's because we are using specific cameras that take advantage of the reflectance of light in different ways to produce these images. So what you see here is the stitching of tens of hundreds of different images as the one that I mentioned before, and each one of these squares is a particular plot in a field that was assembled by the process of collecting these images over a particular season. So this picture over here is the same field. What you see up here is the field of corns in June 17th, probably about a month after planting took place. And what you're going to see down here is the images of normalized difference vegetation index, which is a measurement, again, of the reflectance of the green color, which gives you, based on the difference between the visible light and the infrared light, an idea for the density of green matter in a particular field. So if, let me see, fingers crossed this works. If you look at the left corner, it's showing you the time of the year when these images are taken and how the density of green matter in each one of these plots vary. And you can see that some of the genotypes struggle more with the environmental conditions to the point that you see big holes in the field, either indication of the presence of particular leaf diseases or genotypes that dry faster than the others. And so this kind of information can provide useful indications of the overall health of the plant. So what we wanted to see is whether this type of information correlated with yield, and interesting to see that if we start running correlations between NDVI and yield, we can predict with a significant amount of accuracy the level of yield for each one of these genotypes as early as mid-July. So we are continuing to make efforts on identifying how to best analyze the data and what types of spectral data provides higher correlations earlier in the season. And then the next component of the work that seems to be becoming more and more important is this idea that the majority of the traits that we look at are very complex in nature. So if we can use technologies to dissect those traits into components that would then allows us to look at each of them individually while still maintaining the rate of improvement or the ability to do it in a scalable way. So in corn breeding, there is a significant amount of literature showing how yield, which is a very complex trait, relatively lowly inherited and highly affected by the environment, can be dissected into these different components. So yield depends on the number of kernels on a ear, the weight of that kernel, and then how many ears a particular plant has. And the combination of all these traits provide the overall yield. And these different traits can be further dissected into additional traits. So what we are trying to do is use technologies to try to do that very efficiently and think about how we can do that. So this is National Academy of Science, Ed Buckler, my great colleague, and based on the size and fashion of the sunglasses, you can imagine how many years ago these took place. He does not measure ears in that way at all, and probably he was already using some very sophisticated computing algorithm even then. But we cannot do these kinds of measurements for thousands of ears in order efficiently. This is one of our grad students, and what you see in front of her is the little mountain of ears that she had just out of her experiment in 1 year. So we really needed to figure out ways to do that very efficiently. So what we have done is use technologies such as flatbed scanners that allowed the image of ears and dissections of ears. So this projector is not showing very well, but these image analysis allows us to get measurements of the width of each one of these ears, the length, and then using some relatively simple mathematical tools, we can also determine the number of kernels in a particular ear using just the image. The other interesting thing about this type of analysis is that we seem to always keep going back to these images. They are, the other interesting thing about image analysis is that they are immortalized, and we keep going back to these images and trying to extract more and more information and look at color and contrast and shape to continue to dissect these traits even farther. We do the same thing for cobs, and even for little kernels, the image analysis allows us to now dissect the yield into the three-dimensional components of each of the kernels in a fairly rapid way. And also, one of the really beautiful things about these is that we can, in addition to just measure traits that the ruler can measure, image analysis allows us to do a little bit more. We use principal component analysis on the images taken and reconstruct the most likely shape of particular ears. If you look at this principal component 3, for example, ears when are exposed to stresses towards the end of the growing season, they tend to not form very many kernels at the tip of the kernel of the ear, and that is one known reason for reduced yield potential and under stress conditions. So having access to these types of information can be, for example, an indirect measurement of the effect of different stresses on genotypic performance. And we have done that with other parts of the plants, here briefly describing the work also done with tassels. These type of technologies really allows significant scaling of the effort. We can relatively simply look at tens of thousands of images of tassels in a year and a season. So what is some of the most limiting factors in terms of scaling this technology? So I'm going to use the example of the ear phenotyping to show you the flow of information in our program. So if we look at particular images that are collected on flatbed scanners, really anywhere around the world, those images are pushed to an account, and we take advantage of the NSF-sponsor data storage cybers, the cyber infrastructure, which provides IROD for distributed storage. We just use it really as disk capacity. That information then gets pulled into the high-throughput computing center that is available at the University of Wisconsin. And because of the nature of the type of data that we use, this kind of information lends itself very easily to parallelization of the work. So through that technology, we can run each one of these images through a different computer in real time. And that is incredibly important because of the number of images and the size of the images that we use. Once the computing of the particular images, for example, for ears, are collected, that information is sent back to cybers for storage, and through, again, the high-throughput computing center. So in a given year, we might have 7,000 ears image, about 4 terabytes of data, which will amount, because of the time that it takes to compute, about 10,000 CPU hours. Thanks to these kinds of technologies, such a run could be done in a day. I cannot even think about what would happen if we didn't have access to these kinds of technologies. So we are very happy to have such a technology, but I think we are still very much in the infancies of developing that in a way that would be really scalable just beyond a particular group or a particular effort. So just briefly mentioning how this all is coming together as part of the Genomes to Fields. So the Genomes to Fields initiative is a publicly initiated and led effort, and the idea is to exploit the information, genomic information that has been collected for the last 10 to 15 years to improve our ability to predict phenotypes. And it was really the consequence of, I don't know if I wrote it here, but I think the right word is frustration from those people that had supported the sequencing of so many different genomes that were not seeing how that information was necessarily applied to improve materials. So this effort has been supported to a large extent by different state corn growers and national corn growers associations around the United States. There is a coordinated group, and I am listing it here in case you are part of any of these institutions and would like to learn more. I'm also happy to talk to you more about it. But the idea of this initiative is it sits in a framework that is looking to bring different disciplines here on the square around this umbrella that includes bioinformaticians and computer sciences and crop modelers and statisticians and engineers to create modeling and analytical tools and high-throughput phenotyping tools that can then be combined to get to this goal of improving phenotyping. And one of the first things that we did as the Genomes to Fields group is create a project that would focus specifically on trying to create a platform where all of these technologies, tools could be tested as a sandbox ground to learn what is it that we need to do better in order to move at that scale. So this was initiated in 2014, and what I have here are highlighted in the map are the states where the Genomes to Fields researchers are located. Each one of these states is growing about 500 hybrids, diverse hybrids, across the country and measuring the same traits across all of them, utilizing similar technologies and coordinating on the input and output of data for this entire effort. We do a lot of very traditional agronomic performance, morphological, phenological traits across this collection of diverse materials. And then we are moving now to starting applying some of these high-throughput technologies in places that we can. And we also collect environmental information for each one of these sites through a weather station. So uniform genotypic, uniform environmental, and trying to maximize the phenotypic output from these trials. So just, again, trying to put in perspective what it would take to, for example, bring a ear scanning platform to the genomes to fields, 500 hybrids, about 35 locations in a given year, 17,000 samples to be evaluated. We take 4 images, 2 of ears, 1 of cobs, 1 of kernels. That's about 70,000 images any given year. That's about 20 terabytes of data for just this particular trait. So we need to really be thinking about how we are going to create the infrastructure that will be able to actually handle that. And I've been trying to read how other people have done that, and there is kind of a philosophical difference or a different way of looking at it from people that are more related to the physics and engineering, where I'm looking forward to hear what Victor is saying. But if you look at examples, for example, such as the European Organization for Nuclear Research, that studies very fundamental structures of the universe, how these particles collide in these particular colliders, they have designed their cyber infrastructure before they even collect the first data point. They have simulations, they have pipeline flows that are designed before any data is collected. We are very much doing it as we go, and we are paying the price for that, I think in a very significant way. So the example, they have tiers of analysis where they analyze particular data on the spot before they move it to the next step, before it even makes it to a physicist's shop. These data has been cleaned through automatic processes in a pipeline. And the other thing that is very different is that as complex as the work from CERN is, it is very concentrated, one place in Switzerland, versus something like the Genomes to Fields, which involves several locations across a high geography. So what are the domains of the phenotypic pipeline? So we have a significant amount of work as a community, I think, on acquisition, on sensors and hardware. I think we are doing a decent job of creating the algorithms that generate the results. I think we are still struggling with this last part, which is the modeling. Once you get that data, how to actually use it in ways that would really increase efficiency in the use of these resources. And one of the challenges is that in order to do that, you need to create data standards. Here is the example when we first started in the Genomes to Fields, something as simple as plant height, right? What is complicated about plant height? From the soil to the tip of the tassel, you find out that if you talk to 10 people, you will get 10 different measurements of plant height. So imagine if we start talking about any of the more complicated traits, there is a lot of homework to be done. So with that, I'm gonna conclude by kind of highlighting what I hope I was able to share with you today. So plant improvement has been very successful despite the complexity of the system. But in order to continue that increase, we are required to consider very seriously all of these technologies. The ability to measure intermediate stages in plant development and dissect complex traits into components is becoming really important, not only because we need to increase the prediction ability, but also we need to understand the biology in a more significant way in order to use the information more efficiently. Technologies are becoming available every day. The big challenge is how do we appropriately analyze and interpret these results? And standards on that front become very important, primarily when you start combining information. And the development of an appropriate computing infrastructure is key. I think we are probably a decade or so behind schedule on that. But so I want to thank the Genomes to Fields collaborator. The list is very long. The number of institutions also very large, so I'm not going to go through all of them. Also, the funding agencies that support the work at the University of Wisconsin program. And especially I want to thank Edgar Spalding Group at the University of Wisconsin, my close collaborator, Sean Kepler, the Genomes to Fields collaborator and co-operator, and then this is our crew of grad students, undergrads, technicians. This was past summer. Corn sheds a lot of pollen. It takes a lot of people to get that pollen into the right places. So thank you very much, and if I have time, I'll be happy to answer any questions.
Natalia, you mentioned how it's important to have this multidisciplinary group with the statisticians, computer scientists, the engineering, and the agronomy people. So how difficult do you find to attract people outside the agronomy, colleges of agriculture for people to collaborate with us? How you built this big group, for example?
You know, it's not difficult to attract them. It's easy to establish a conversation and get very excited about it. Engineers are always looking for problems, and we have so many of them that it is actually not hard to get them excited. I think one of the hardest things is to identify funding to justify investing the effort. So thinking about how do we create a funding infrastructure that would foster that, I think it's critical. In that I think it's not only critical, but I think it's also, it's really a waste of resources. There are so many things that we are doing that have been done before in other disciplines. If we just had a chance to know about that, it would be really awesome.
You spoke of the challenge of modeling, and I guess from a dairy cattle breeding perspective, we've made great progress in genomic prediction. Is that the goal to be able to identify lines or that you should cross and identify the most promising crosses? Is that the goal that you see in being able to do this modeling?
That's exactly right. So, and I didn't have time to go into the details, but some of the things that we are working on is we are collecting all this information. How do we actually incorporate that information into prediction models? We have a ton of, we have marker information, we have expression information, we have metabolomic information, and now we have this slew of measurements of phenotypes over a season. How do you actually combine all that information and improve your efficiency to select those superior individuals? And the other question is also, what traits are actually adding information to what is already there? And that is also very maybe specific to the specific situation. So it's really there where I think people that are investing energy on thinking about ways to improve that process, that is really incredibly critical. Yep.
So another question here about when you talk about the traits, do you look at the biochemical traits also? Like, for example, how nutritious it is, like a lot of what you're producing is going to the animals. So do you work with any of the nutritionists to see how you can improve, the quality of the feed so that it can be, better used?
Yeah, so I already, my presentation went pretty long. So so my day job, I'm a corn silage breeder. That's part of my responsibility at the University of Wisconsin. So we do a lot of work using technologies such as NIR and things like that to improve the efficiency and understanding composition. I mean, the list is endless. Each one of those could become an important technology depending on what the outcome that you're interested in. So, I mean, most definitely, if instead of having to collect the biomass from a silage sample, grind it, run it through the NIR. If I could just go out there with a LiDAR detector and just touch a leaf and be able to figure out the composition of that leaf in a way that would be meaningful for when I harvest an entire corn plant in terms of the composition, ruminant composition, that would be amazing, right? I mean, that is how we make the breeding process more efficient. Yep.
Hi.
Yeah.
As we're trying to sort of anticipate some of the problems we may be faced by such high-throughput phenotyping, can we learn vicariously from perhaps some of the mistakes you've made?
How much time do you have?
Some of the critical points that you would advise us to keep in mind?
I mean, I think the issue of standards is a very serious one. And it is tricky because you also don't want to box people into always measuring things in the same way. But in order to really exploit data and make data useful across programs and across efforts, there has to be a way to make things more uniform, at least to a certain level. Also, I think one of the things that we maybe underestimated is the issue of how this dispersed nature of our effort becomes challenging in the sense of moving data in and out from the different sites into a common computing infrastructure and then bring the data back out. They don't seem like overwhelming issues, but from the standpoint of implementing the genomes to fields, at least, those have been sort of the first roadblocks that we have faced. The third one is just getting enough funding to get everybody to do things at the scale that we need. But that is kind of, everybody suffers from that problem.
Did you find Cybers a useful way to solve that second problem?
So Cybers is great. I think Cybers is Cybers is an NSF-funded effort, and therefore it is free to researchers, which it's not a minor thing. They have expertise that can help in terms of the storage component of the effort. They are very good at that. The computing capacity or the tools that are available, I don't think they were at least not useful at the scale that we needed. And that's where I think it is important to consider maybe exploring other possibilities or thinking about what people have done in the physics and engineering, for example. Yeah.
Natalia, can you say a little bit about data sharing for the project?
Yeah.
Because if you have some researcher from MIT wanted to come in, how would they partner with the project?
Thank you. I'm sorry, I should have mentioned that. So for the Genomes to Fields effort, the data that is collected becomes available to the co-operators of the effort basically immediately after we collect it. We curate it very minimally to make sure it's not completely out of whack, and then we make it available to co-operators, and then that data becomes publicly available about a year after that. So the 2014-15 data, genotypic, phenotypic, environmental data, is currently publicly available. 2016 will become available in March of this coming year, and the idea is for all this information to continue to be available, whatever traits, anything else that is measured. Yeah, thank you for that. Yeah.
So I have a quick question. So you talk about genetics by environment, but one thing you didn't mention is management. And we have, in the animal world, we can manage animals in a whole bunch of different ways. And in the corn world, I would guess that you have fertilizer, you have irrigation, you have rain amount. How do you take that into account?
I should have listed that on this gentleman's question about the first struggles. It is hard. So, definitely when I'm talking about environment, I am thinking about the combination of environment and management. That was certainly one of the challenges for the genomes to fields, is because all of these are these investigators have received a little bit of seed money from their own state corn growers, but it's relatively little, and they are leveraging their own resources. So we all have different combines. We all put different fertilizer. We all have different irrigation or not systems and so forth. And so we are trying to incorporate that through modeling and using whatever covariables we have from the weather stations for the analysis, but it is far from perfect. If we could design these from scratch, it would be my dream come true, just ship a same combine across all these 35 locations so we actually have the same data. That would be quite incredible. Very difficult though, $350,000 a piece, can you imagine? So, but yes, definitely, that's definitely something that we have to think about and be very aware of all the time. Yeah.
If there's no other questions, let's thank Natalia for a wonderful start to the conference.
Thank you so much.